The very last part of Chapter 11 deals with an interesting cellular activity called apoptosis or apoptosis. I looked up how to pronounce it, and I got like 4 different variations. So feel free to pronounce it however you wish, but it looks like apoptosis or apoptosis, and it says it integrates multiple cell signaling pathways. And this particular pathway is basically programmed or controlled cell death or cell suicide. A very interesting thing that occurs with cells where cells basically go through a process where they commit suicide, and the interior of the cell is chopped up by different enzymes and packaged into vesicles that are then digested by scavenger cells. And this is actually a good way to go about this, because remember, the cell contains numerous enzymes that could be harmful if they were just leaked out onto the other cells. So when a cell is ready to undergo its death or its suicide, it goes through this apoptosis, which basically chops up the cell on the interior and then creates these vesicles that surround all the material, and then those vesicles are basically eaten or taken in by other cells. And that way it prevents any materials or enzymes or infectious materials from leaking out of the dying cell and damaging living cells around it. Apoptosis comes from a Greek word that means falling down, and originally it was used, I believe, to talk about leaves falling from trees, like in the fall when they go through senescence. But now it's primarily used as an explanation or a word that means cell death or pre-programmed cell death or cell suicide. So cells that are infected will go through this process. Sometimes cells that are damaged in some way, sometimes the DNA gets damaged or organelles get damaged, or simply cells that have basically reached the end of their functional lifespan. They'll undergo this process called apoptosis, and that is involving numerous kind of cell signaling pathways to get that process to go through. There's a very good video on YouTube. You can click on the link right here and go to a YouTube video where you can see kind of a live shot through a microscope of apoptosis going on, and it's kind of interesting. The cells basically just sort of disintegrate in front of your eyes. They shrink down, and then we'll see they actually kind of change shape, and they look quite a bit different than they did when they were alive. Here's apoptosis of a human white blood cell. The normal looking living white blood cell is on the left, and then the cell that's undergone apoptosis is on the right. This is called blebbing, kind of its nickname, where it forms all these little bubbles or vesicles, and the cell inside has basically been gone through a blender. It's been chopped up by all sorts of enzymes that chop up the DNA and chop up all the proteins, and then it forms all these little vesicles. So kind of an interesting word. I don't know where it came from, blebbing, and that's kind of the process of apoptosis. Because of its association with the cell's death or kind of the programmed cell suicide, apoptosis would generally be thought of as the end of the road for a cell, and that would be kind of a negative connotation. But actually, apoptosis is also very important in the embryonic development and the shaping of the organism while it's still in the egg or still in the womb or still developing prior to its birth. So human embryos, when they are forming into a fetus and then eventually into an infant, they go through apoptosis, and most living creatures do as part of their embryonic development. A good example of this is in a particular soil worm. It's got a long scientific name here, which I'm not even going to pretend to know how to pronounce, but we're just going to go with C. elegans. C. elegans. It's a small soil worm, a nematode, which tends to be very tiny. Oftentimes they're almost microscopic, so you can't really, not like an earthworm that you could just pick up and hold in your hand. They tend to be very small, only about 1,000 cells when they're completely grown. So we're talking about a pretty small creature here. But because it only has about 1,000 cells, scientists use this particular creature to kind of study this embryonic process and this apoptosis process, and they could pretty much track all 1,000 cells and kind of know where they came from and the various procedures they went through. So in this particular worm, apoptosis results when there's a particular protein that accelerates the process. So the interesting thing is the protein or the enzyme that's in there to chop up the cells and cause this death, it's always there, but there's another protein that kind of holds it in check. So when that check protein is kind of taken away or inactivated, then the other proteins that were already there to begin with, they can do their job and begin the whole blending apoptosis process. So take a look at this particular model here. You have a living cell on the left, and inside here you've got these two proteins. They're called CED, CED4, and CED3. They're inactive at the moment, but these are kind of like the death proteins that cause the apoptosis, that do the whole chopping up of all the other proteins inside here. So these are enzymes, and they're inactive at the moment. And the reason they're inactive is because this protein, CED9, is active, currently active. So it's kind of like the check valve that keeps these two from going crazy and doing what they want to do. Now, when the cell receives a particular signal molecule, so here is what they're going to call the death signaling molecule, that comes into the receptor, and then that activates a whole network of steps that then inactivates this particular enzyme. So then this particular protein or enzyme gets inactivated, which then allows these two to become activated. That begins a whole process of proteases and nucleases. Proteases and nucleases are the enzymes that chop up various other proteins and nucleic acids, and that begins to destroy the cell. In the meantime, the cell begins to form these blebs, these little bubble vesicles. All the chopped up parts are contained within the blebs, and then those blebs are basically engulfed by other cells. So when this inhibitor protein is inactivated, that allows these other proteins to become active, and they start the whole process of destroying the cell. Together, these are called caspases. Caspases are basically the ones that do the scissoring action, the chopping up of all the various proteins. So proteases would destroy proteins, nucleases would destroy nucleic proteins and nucleic acids, basically cutting them up into small little pieces and then forming them into the blebs. So within the cell, there are these caspases that are always there. They just aren't activated until some particular signal is received. So the caspases are the main proteases, which are enzymes that cut up proteins that carry out this process of apoptosis. In humans and other mammals, there's several different pathways that involve probably altogether about 15 or so of these caspases that can carry out apoptosis. The particular caspase that goes to work to destroy the cell kind of depends on which particular signal molecule is received and which pathway, and that kind of depends on which cell type that we're talking about. So apoptosis is usually triggered by some sort of extracellular ligand, so some sort of external signal that the cell receives, like we saw in the previous example. And it can also be triggered by DNA damage. So there are some internal signals that cause this. So if the DNA becomes somehow damaged in the copying process or through basic cellular processes, that oftentimes triggers this whole apoptosis process to occur. And then also if we have too much protein malfunction. Remember we talked about the endoplasmic reticulum, where the proteins are formed and then folded into the very complex shapes. Well, if we have a problem where the proteins are being misfolded, that can cause a cascade of events that will result in the cell saying, uh-oh, we're too far gone, we're too damaged, and then it goes through this apoptic process. That's a hard word. Apoptotic. Oh, I don't even know if I can say that. Apoptosis, apoptotic pathway. So if it gets to this point where it realizes, and when I say realize, of course, I don't mean it has a brain and it's thinking. Certain signals are sent out that says the DNA is damaged or the ER is damaged in such a way that it's misfolding proteins. It'll start this effect and will end with the suicide or the death of the cell. If we look at the genetics for creating the various enzymes, the proteases, and the various materials involved in apoptosis, these apoptosis genes are seen in basically everything from nematodes to mammals, multicellular fungi to single-celled yeast to lots of living creatures, pretty much all living creatures. If we look at the genetics that determine how this apoptosis process occurs, it's very, very similar. So again, it's thought that this whole process evolved very early in animal evolution, and it's pretty much essential for the proper development and maintenance of all animals. You have to have cells basically discarded at a certain point. And with embryonic development, you have to have programmed cell death for the creature to be formed in the proper way. So while it's kind of a negative thing, obviously, for that one particular cell, the process of apoptosis is essential for the normal development of the creature, for the development of things like the nervous system, and for the normal functioning of the immune system, and then obviously for the normal development and formation of things like hands and feet and paws. In the whole embryonic process, you have to have apoptosis occur. Also, in modern medicine, we've kind of come to realize that apoptosis may be somewhat related or have something to do or involved with certain diseases like Parkinson's disease or Alzheimer's disease. And we do know that oftentimes different cancers kind of interfere with this apoptosis process, because normally speaking, when cells become cancerous, they begin to reproduce out of control and they're damaged in some way. Normal cells would go through apoptosis, and that would limit the damage. Cancerous cells tend to have a way of kind of shutting off this apoptosis process, and so the damaged cells continue to reproduce. So that is a major part of certain diseases like cancer. The signaling pathways that kind of control apoptosis and feed into this are very elaborate, and there's a lot of different steps, and that kind of makes sense because it's probably the most important process that the cell might go through. I mean, it's a matter of life and death for the cell, so it makes sense that it's a multi-step process before the cell kind of determines that it's going to commit suicide or this is going to happen. Again, I don't want you to think that cells think for themselves. Of course they don't. A cell can't think, but the entire chemical reaction process that occurs is very complicated with multiple steps. Let's take one good look, last look at apoptosis kind of in a more positive light, and that would be with the formation of digits and things like that. So this is with a mouse, but the same thing occurs with a human hand. If you looked at a mouse embryo very, very early in its development, in a human embryo very early in its development, the hand doesn't look much like a hand or a paw at all. It looks like a web, more like a duck's webbed foot or something to that effect. You can see some little bumps, which will become the digits later on. And then what happens is, as that embryo continues to develop, you have cells that start to undergo apoptosis. And the lighter cells here are marked in such a way that you can see where apoptosis is occurring. So those cells actually go through pre-programmed death in between the spaces until eventually you end up with digits 1, 2, 3, 4, 5. So even though this is a mouse, it could be human at this point. It looks very similar. And then you have further development into a mouse's paw or a human hand or a human foot. So apoptosis is not always a bad thing. It's very integral to the development of animals in the embryonic stages, and it's a part of keeping the whole system working properly, but it's definitely a mechanism that requires a whole lot of cellular communication using all these pathways that we talked about. We're going to stop right there. That's Chapter 11. Be sure and take a look at the textbook. Go online and take a look at any of the videos I might have shown you. There wasn't a crash course video about cellular communication. I will go and see if I can find something else, but I would rely on your textbook and our vodcast right here, and I think you'll do fine. Talk to you soon. Bye-bye.